Hi, my name is Diego Castillo, and this is humoral immunity. The phases and types of humoral immune responses. The first part of a humoral immune response begins with antigen recognition, which then will activate the B lymphocytes. Once activated, the B lymphocytes will proliferate and differentiate, eventually leading to antibody secretion, isotype switching, affinity maturation, and the formation of memory B lymphocytes. There are two phases of humoral immune responses, with the primary immune response, where the immune system encounters an antigen for the first time. There's a lag phase that will occur where B cells are activated and produce antibodies, but it takes them a minute because they've never been exposed to this antigen. This response is much slower and not as effective as it would be if it were exposed to the same antigen. The secondary immune response, where the body reacts to the same antigen again, and memory B cells recognize the antigen, producing antibodies faster and in more amounts. Since there is memory, the response is much quicker and more effective, and it will produce a better protection. So the stimulation of B lymphocytes by antigens. There's two types. There's T-dependent and T-independent. For T-dependent stimulation, in the case of T-dependent antigens like proteins, follicular B cells will bind to an antigen through the BCR and engulf it. Once processed, it is then presented on the cell surface through MHC class II molecules. Helper T cells will bind to the MHC class II and will recognize the antigen. This will allow the helper T cells to release cytokines that will stimulate the B cells, allowing for the B cell to proliferate, differentiate, and produce antibodies. There will be clonal expansion and differentiation into plasma cells and memory B cells. The main results will be isotype switching in order to respond correctly and produce high-affinity immunoglobulins that mature. For the case of T-independent antigens like polysaccharides, lipids, and non-protein antigens, will stimulate antibodies without the need of T helper cells. It is useful because there is cross-linking of the B cell receptors and that they can activate the B cells strongly enough to stimulate proliferation and differentiation without T helper cells. Mainly, IgM is produced, which are low affinity but high avidity, as well as plasma cells that don't live long. Similar to T-dependent activation, there will be clonal expansion, differentiation, and antibody production. But it differs because immunoglobulin M is produced instead of IgG, IgE, or IgD, and since IgM is more high avidity, where it has 5 chains, it is not as high affinity as other molecules or antibodies. The response is rapid, but it is shorter lived, and there is a lack of formation of memory B cells, so there won't be any long-term immunity. So the role of innate immune signals in B cell activation. The complement system will activate and lead to the binding of a complement breakdown product, C3D, to microbes. The B cell will also simultaneously recognize the same microbial antigen and bound C3D. CR2 will be attached to a complex of proteins that are involved in sending activating signals to the B cell. Pathogen-associated molecular patterns can activate TLRs of B cells simultaneously as microbial antigens are being recognized by the antigen receptor. So B cell activation will occur due to many of these different interactions with the cell, and it'll activate the adaptive immunity and B cells themselves. So heavy chain isotype switching or class switching. So helper T cells will stimulate the progeny of immunoglobulin M and immunoglobulin D. B lymphocytes will produce antibodies with different heavy chain isotopes. This class switching will be induced by CD40L-mediated signals and cytokines. Cytokines produced by follicular helper T cells will decide which class isotype is produced. Each class of isotypes is different in use, with IgM being mainly to the first responder of the immunity, where it'll recognize different antigens. IgA is mainly through mucosal immunity. IgE is primarily against helminths and for allergies. And IgG is the most prevalent in the blood, and it usually is responsible for Fc receptor dependent phagocyte responses and complement activation, as well as neonatal immunity.
Affinity maturation is the process where we undergo mutations to enhance and empower the effectiveness over time. The main goal is for the immune system to allow antibodies to switch up and change their binding to antigens, this leading to a more effective and specific immune response. This process happens primarily in B cells during an immune response, especially after exposure to an antigen through infections or vaccination. It starts with the activation of B cells, where a naive B cell meets an antigen and becomes activated. Then the cell's DNA undergoes mutations, creating a different version. Then the mutated B cells compete to bind to an antigen, and the B cells with higher affinity survive, and then eventually divide, this process being called clonal selection. The main goal is to produce stronger and more specific immune responses. Regulation of humoral immune responses, antibody feedback. Antibody feedback is a crucial role that helps regulate the humoral immune response in the body. It does so by preventing the overproduction of antibodies. Once antibodies are produced, their modulation of them happens in order to affect future B cell responses, in order to enhance or inhibit them when needed. By clearing antigen blocking epitopes and engaging inhibitory Fc region receptors, antibodies can have a negative feedback effect that may limit the magnitude of the response. IgG antibodies bind to leftover antigens, this forming antigen-antibody complexes that will then interact with B cells. The FcYRIIB receptor is the one that is attached to the B cell. Effector functions. Neutralization of microbes is what happens first, and microbial toxins. This happens when antibodies are binding to and blocking or neutralizing microbes and interactions. These antibodies are found on epithelial lining and mucosal entrances like the GI tract and act as the first line of defense. Opsonization and phagocytosis happen when there's a coating of the microbes in order to promote phagocytosis. Ig subclasses bind to microbes and are distinguished by Fc receptors. The third step is antibody-dependent cellular cytotoxicity. This is when NK cells bind to antibody-coated cells to destroy them. Antibodies bind to infected cells. The Fc region is recognized by the Fc receptor on these cells. The fourth step is IgE and eosinophil mast cell mediated reactions. This happens when IgE activates mast cells and eosinophils. This provides defense against helminths as well as allergies. Finally, the activation of the complement system takes place involving all three pathways: classical, alternative, and lectin pathway. These are triggered when IgM or IgG subclasses binding to antigens, this being the classical pathway, or triggered by complement pathways, which is the alternative pathway. The lectin pathway, however, is not initiated by any antibodies. Vaccines. Vaccination is a key function in immunology. It stimulates an adaptive immune response as well as it aims at preventing not only the infection but also the spread of the disease. Vaccines serve as a type of long-term immunity to either protect or weaken from the disease so the host is not affected so harshly. There are numerous types of vaccines, starting with the attenuated vaccine. These use weaken microbes that can infect but not cause disease. There is also subunit vaccines that contain only specific proteins from the microbe itself. Conjugate vaccines are those which link polysaccharides to proteins in order to help activate helper T cells and enhance the antibody production. Synthetic vaccines are simply in the name. They use lab-made proteins. Viral vector vaccines are those that develop microbial genes using harmless, not bad viruses in order to trigger an immune response. Lastly, there are DNA vaccines that involve injecting DNA into the host cell in order to produce antigens and start triggering and stimulating an immune response. Thank you.